Humans and the microbial world. This first part will look at the study of microbiology, the definition of microbiology, definition of germs. We will look briefly at the study of microorganisms from a historical perspective, that is, the history of microbiology. We will look at the contributions of Hooke and Leeuwenhoek, the theories of spontaneous generation versus biogenesis. We will look at Pasteur's gooseneck flask experiment and its significance. We'll look at Tyndall's contribution, and finally we will explore a few terms important in the laboratory growth and study of microorganisms. By definition, microbiology is a science that studies living things that are too small to be seen by the unaided eye. Thus, micro means small, bio means life, logi, the study of. Having said that, microbiology does study living things that can be seen with the plain eye. For example, algae are one of the microorganisms studied under this discipline, and it is not necessarily a microscopic living thing. If you look under the microscope at pond water, you would be amazed at the number of microorganisms that can be seen and the different types of microorganisms. These large green structures are the algae. You may see slightly smaller protozoans and tiny bacteria. This tells us two things about microorganisms. First of all, they come in many different types and sizes, and they are found in the natural world, all living together. So, three different types of microorganisms we have already mentioned will be protozoans, algae, and bacteria. And as you can see, they are not all alike, and all of them coexist together in their natural. You may have heard of the term germ. Germ is used to refer to a disease-causing microorganism or microbe. Note also that throughout this course, we may use the shorthand MO to refer to the word microorganism. You should know the meaning of the term germ. So at this point, you should be able to define microbiology, define the term germ, and give a list of some of the organisms, microorganisms, studied by microbiologists. So are all organisms studied by microbiology microscopic? Well, not really. That you have microorganisms with your plain eye. Now, the study of microbiology is intimately related to the development of the microscope. Without microscopes, we wouldn't have microbiology. The scientist who is credited with developing microscopes is Robert Hooke. He's an English microscopy of the middle to late 1600s. He published a book called Micrographia, where he describes how to make microscopes, and he reports seeing some microorganisms like the common bread mold. So we have all seen microorganisms in our daily life. We may not have realized they were microorganisms. For example, we've all seen moldy bread, but if you take that moldy bread and look at it under the microscope, now that you see the actual structures of the mold, you can see the filaments, which are called hypha. You can see the round structures at the end of the filament, which are the fruiting bodies that carry the mold spores. However, the really small microorganisms, the ones that can only be seen by the naked eye, were first reported by a Dutch cloth merchant named Antony van Leeuwenhoek, which was a contemporary of Hooke. He discovered microscopes also, in the sense that he began making his own microscopes after he read of Robert Hooke's work, and he became a very good microscopist. He initially used microscopes to look at the quality of the cloth in his shop. He was not. He very soon began looking at other specimens under the microscope, things like blood and saliva and pond water. So he is the first one to actually have reported and recorded seeing microorganisms. These are some of his drawings in which he described these little animalcules, as he called them. And this is the actual microorganism under a modern microscope. And you can see that the difference is not, they're not very different. The similarities are quite striking. He was a very good, very detailed scientist, even though he was just an amateur scientist. These are some of the microscopes that he made, and you can see they were very simple. The main component of it was the lens, and that's where the secret of his success in microscopy lied, is that he made very good lenses that were able to increase the size of a specimen by up to almost 300 times. So that was a good magnification for that time. So Van Leeuwenhoek recorded and reported the existence of microbes. Now, it took him a while to convince the scientific community of the existence of microbes. But once the concept was embraced, the next question that scientists now began asking is, where do they come from? Where do these little microbes come from? It was clear their existence, and they could easily identify their presence. For example, when there were very few microorganisms on a broth, the broth was clear. However, as they become larger in numbers, the broth turns cloudy as it is teeming with microbial life. So a dispute came about trying to explain where do these little microorganisms came from. And the two theories that tried to explain this concept were the theory of spontaneous generation and the theory of biogenesis. The theory of spontaneous generation explained that these small living things came from non-living materials such as air or water. And this theory was arrived at through numerous experiments that appeared to show that microorganisms came about from non-living substances. On the other hand, the theory of biogenesis reasoned that all living things must come from other living things of their own. So there were numerous experiments trying to support both theories, the spontaneous generation theory, and you can read about some of these experiments in your textbook. By the late 1700s, the theory was still raging and there was no real resolution to it. However, spontaneous generation by then had concluded that in order for non-living things to bring about life, there had to be a living force in the non-living matter. This force may be destroyed by boiling, which meant that boiled broth or boiled heated air would not have the living force that made possible the appearance of microorganisms. So again, living things did not appear through substances that had been heated or boiled because the living would have been destroyed. So the controversy raged until the late 1800s, when a French chemist named Louis Pasteur took on the project of disproving spontaneous generation. He designed an experiment that is now called the Goose neck flask experiment. You can read about the experiment in the textbook. His challenge was to prove that there was no vital force in the air, that the source of microorganisms was other parent microorganisms. The way he designed his was that he created these special flasks that had a long curved neck. He used beef broth as the source where microorganisms would be growing. He took the beef broth and he boiled it. Now remember, according to the spontaneous generation theorists, once you boil the substance, now the life force is destroyed, therefore no microorganisms will spontaneously arise. Well, in his experiment, the gooseneck flask was left open, which meant that air that had not been heated could go into the flask and repopulate the broth if the air indeed was the source that made possible the presence of microorganisms. However, if the air was not the source of microorganisms, there was no such thing as a vital force, then the broth will remain microbe-free. What Pasteur theorized is that the air was not the source of the microorganisms, but rather the dust carried by the air was carrying microorganisms. The dust, however, was being trapped by the curves in the flask. So the purpose of the curves in the flask was to trap the dust that was carrying microorganisms. Therefore, what was reaching the broth was filtered air. The broth remained microbe-free until the flask was tilted and the broth was allowed to come in contact with the dust. Then the broth showed microbial growth. These showed that it wasn't the air the source of the microorganisms, but rather other microbial cells that were caught in the dust. The open flask allowed for fresh air to move in, proving that the air did not have a vital force. The curves of the flask allowed for entrapment of the dust carrying microorganisms. So the experiment supported the theory of biogenesis. Thus, Pasteur is called the father of microbiology. He was actually a giant of his time. He worked in other projects related to microbiology. He disproved the theory, not only disproved the theory of spontaneous generation, but he also worked on fermentation and proved that fermentation was due to the presence of microorganisms. He designed a procedure to control microbial growth in food products, which now we call pasteurization. He also developed the first modern vaccine, which was the rabies vaccine. So Pasteur's gooseneck flask experiment suggests to us some of the basic microbiology tenets. It shows that microorganisms are everywhere. They were in the air, in the dust carried by the air. The experiment also showed that we can see microbial growth because microorganisms growing in clear sterile infusions will over time make the infusion cloudy when there are enough of them. It also showed that a sterile infusion will remain sterile for as long as they are preserved free from microorganisms, which means that growth of microorganisms can be controlled by heat. Remember, Pasteur was heating the broth to destroy the microorganisms already present in the broth, and the flask remained microbial free as long as the dust was not able to reach it. So if microbial growth can be controlled, then we can study microorganisms in the laboratory. So very soon, scientists like Pasteur and Koch and Tyndall and others devised methods that allow them to control microbial growth. These methods are called aseptic techniques. Aseptic techniques are very, very important. This is what allows to control the presence of microorganisms on an environment. You should know the definition of aseptic techniques. We will use aseptic techniques in the laboratory all the time. Aseptic techniques allows microbiologists to isolate, culture, identify, or study microorganisms. To isolate microorganisms means to take them out from their natural environment into a control environment, like a laboratory. To culture microorganisms means to grow them in a controlled environment, like in the laboratory. To identify is to know what kind they are, and a study is to learn about how to keep them alive and what kinds of metabolic processes they are capable of. Again, aseptic techniques are a very important set of techniques that makes it possible for us to work with microorganisms in the laboratory. By definition, these are techniques, methods that allows us to exclude contaminating microbes from an environment. We use aseptic techniques also in the medical world. During a surgery, we use aseptic techniques to keep an environment sterile. During medical procedures, like catheterization or the use of respiratory devices, we use aseptic techniques to exclude possible contaminating microbes from a clinical environment. Now, going back to the gooseneck flask experiment produced by Pasteur, once an experiment is put forth, it is expected that other scientists can conduct the same experiment and obtain the same results. So many other scientists began to trying to reproduce the gooseneck neck experiment with mixed results. Notably, a medical physician called Tyndall, English physician, he tried to reproduce the gooseneck flask experiment with very little success. He finally realized that different infusions, different types of growth mediums require different boiling times to be sterilized because some forms of microbial life were heat resistant. This is actually a very important concept of microbiology. Essentially, what was happening with his experiment is that instead of using beef broth, Tyndall was using a hay infusion to create the medium. The problem with a hay infusion is that it has dirt in it. Dirt oftentimes contains bacteria that are capable of producing heat-resistant forms. So he finally realized that the problem laid in the hay infusion and the dirt bacteria found in the hay infusion. So dirt contains bacteria that are called endospore-forming bacteria. So these are endospore-forming bacteria. The bacteria can have two forms of life. One form of life is a normal cell, which is heat sensitive, and he called it a vegetative cell. However, when these vegetative cells were exposed to an environmental stressor like heat, which is what he was doing with his hay infusion, these vegetative cells changed into a heat-resistant form, which he called an endospore. Once the environmental stressor was removed, the endospores came back and turned back into a vegetative cell. So he discovered that some bacteria could produce a heat-resistant structure, making it much more difficult to control their presence and their growth. So a sterile solution will be one that has no forms of microbial life present, including endospores. So now scientists realized that they had to increase the heating temperature and time in order to get rid of endospores. Solution sterile. We will see endospores in the laboratory. We will work with endospore-forming bacteria and stress it to produce endospores, and then we will stain a sample to see the vegetative cells and the endospores that are formed in response to the stressor. So at this point, you should be able to name and contrast the two theories that sought to explain the origins of microorganisms. You should be able to explain how Pasteur's gooseneck flask experiment disproved the theory of spontaneous generation. You should be able to list the 3 points regarding the microorganisms that can be inferred from Pasteur's gooseneck flask experiment. You should be able to define aseptic techniques and know the 2 types of bacterial life that were found by Tyndall. Now remember that Tyndall found its endospores, the endospore-forming microorganisms, in hay infusion, which had dirt, which contains the endospore-forming bacteria. What would have happened to Pasteur's gooseneck flask experiment if he used his infusion from hay instead of broth? Well, he wouldn't have been able to produce an experiment that worked the way he had theorized. Remember that we mentioned the microorganisms growing in a broth make the broth cloudy once there are enough of them. So can you tell which broth has more microorganisms growing? Well, clearly A, which is a lot more cloudy than B. That doesn't mean B has no microorganisms. We don't know that, whether it does or it doesn't. Now, if we say that solution B has no microbial growth, if it has been sterilized, then you can say, okay, then if B is sterile, then it has no microorganisms. So if a sterile solution is one that has no microbial growth, including endospores. this concludes this first set of PowerPoints of Chapter 1.